The overall goal of this procedure is to analyze the in situ sulfur isotopic compositions of various pyrite generations by secondary ion mass spectroscopy in order to understand the diagenetic history of pyritization in methane-bearing sediments.
This method can help identify the biogeochemical processes that impact pyritization, like organic clastic sulfur reduction and sulfur-driven anaerobic oxidation of methane, which cannot be resolved by bulk sulfur isotopic analysis.
The main advantage of this technique is its high resolution and precision, which allowed us to analyze the sulfur isotope composition of a pyrite on microscale.
Demonstrating the SIMS analysis procedure will be Zhiyong, a technician from the SIMS laboratory at the Guangzhou Institute of Geochemistry.
To begin, clean the surface of a sediment core and collect a set of samples across the entire length using a knife. Pack the wet samples individually in zippered plastic bags and label them using a marker. When ready to continue, transfer the wet sediment samples into pre-cleaned beakers and place them in an oven at 40 degrees Celsius for 24 hours to dry them out. After drying, separate the sediments into two aliquots, one for the collection of pyrite aggregates and the other for bulk sulfur extraction. Add 400 milliliters of distilled water to soften one aliquot of the sediment for 2 hours. Transfer the slurry into a .063 mm sieve and sift the sediment with distilled water so that all fine grains are washed through the sieve. Collect the coarse fraction in beakers and dry them in an oven at 40 degrees Celsius.